soccer player. I think there's a soccer player, James McCarthy's on, probably the least cool reader I know. Anyway, yeah, thanks for the introduction. Lots of co-authors on this talk. This goes to show how much of a collaborative effort this was, with a lot of people helping over a long time. So thanks to everyone that's here. So the work that I'm talking about was published earlier this year in Biological Conservation. It's open access, so you should be able to find it if you have a little look. Otherwise, you're welcome to get in touch, and I can send a copy through. So yeah, Lophomyrtus bullata. This study was focused on patterns of Lophomyrtus bullata. It's a Latin name, so the name is rama, and it's infection by myrtle rust or influenced by environment and disturbance. So notionally, it's a tree that gets to sort of around 9 or 10 metres, but I've actually seen individuals much taller than that. It occurs across the North Island and also the North and South Island. And it has a really patchy sort of scattered distribution. People might have noticed this, you come across a patch of it really next to them, and you'll go somewhere else that seems otherwise identical and it's not there. We think this is because of its, the way it's dispersed by birds. There's a fleshy fruit that birds like. It's a shade-tolerant species, but it's responsive to light. And I've got down the bottom there, these slides are a little bit old, that it's probably the most susceptible species to myrtle rust in New Zealand. But it sounds like Myrtle is definitely a bit of a crop for its money, but probably in the top 2. So this study started with us hearing about observations of higher infection of Lophomyrtus bullata closer to forest edges. And this sort of made sense to us. You know, at forest edges, ecosystems tend to be less intact, they're more disturbed, and these forest edges might also provide an entry point for wind-grown myrtle rust spores to enter and infect individuals. And there's quite a big literature looking at edge effects in forests, often from sort of a conservation perspective, and generally people find that edges tend to be more disturbed, more weedy. Native species that you like may not be there, and as you get further into the forest, you start hitting these more sort of pristine environments, fewer weeds. That's kind of the paradigm that we approach this study with. So we decided to set up a study to look into it. So it looked something like this. We established these 60 by 10 metre transects, each with 24 5 by 5 metre subplots running from the edge of the forest up into the towards the interior. And along the centre of these transects, we had 4 temperature and humidity sensors, which you can see. These are these sort of blue dots that I've got on this diagram. And within each of these 5 by 5 metre subplots, we measured the basal diameter and the height of every Lophomyrtus bullata individual that we came across. We looked at plant community composition using we call them recce methods, but basically everything gets sort of identified and counted. We looked at the percentage canopy cover using a leaf area index device, which sort of tells you how shaded the area is based on the plant cover above. And then we also tagged 3 Lophomyrtus bullata individuals for further monitoring. And these were the tallest individuals between, so they had to be greater than 30 centimetres tall and less than 2 metres actually. Some individuals were better than 2 metres, but we weren't able to do our assessments reliably on plants that were taller than those, so we just sort of kept ourselves within that size range, and we looked at the disease load on those individuals over time. So we established 3 of these transects in Mount Taranaki on the southern side of the mountain, in an area where we knew there were a lot of Lophomyrtus bullata. It's actually quite hard to find places where you can implement an experiment like this, where Lophomyrtus bullata is sort of consistent enough along these forest edge gradients. We'd like to have repeated this at multiple sites, but just couldn't find places where we could repeat this. And this is where we knew myrtle rust was present as well, and it likely had been for a year or 2 prior to this. So yeah, 3 transects established. That's a total of 72 subplots and 145 tagged Lophomyrtus bullata that we looked at. So this is an image of the understory of one of our plots, sort of typical of the forest we're working in. It's a relatively damp forest, sorry, that's spiraling, with a moderately sort of dense understory as well, such as tree ferns, kiki, and a wooden overstory of Kaikatea, Pukatea, Tawa, and so on. So really nice forest. There are other myrtaceae at the site. You can see some of the climbing rata on that central tree there in the background. And there's also Syzygium Maritawaki trees at the end of one of our transects as well, where the environment sort of started getting a bit wetter. So our disease assessments, we did 3 of them across 3 years, February 2021, 2022, and 2023. So this is when we returned at the time that we were relatively certain was peak infection. This was based on some pretty intensive monitoring on the plant sort of nearby to the site. So we're pretty confident that we picked the time of peak infection. And we quantified disease on those tagged individuals that I told you about before. So we inspected every single leaf. And first, we scored whether they were immature, or whether they were dead. And this is important because theoretically it's only the immature leaves that can be infected by myrtle rust. Once they sort of harden and become mature, they become immune. We then assessed whether they were infected or uninfected. We didn't do any quantitative measure of how infected they were. It was just a yes or no. And then we also inspected immature stem sections and buds, which can also which are also susceptible to the disease. And as I said before, 3 years, we did this over 3 years to look at the environmental drivers of infection and also how this changes over years. So this is just the first result I'll show you. I'll show you this is just the number of leaves that we counted across those 3 years. So in 2021, there were a bit over 35,000 leaves that we inspected. And immediately, the second year that we went, there was a big decrease. We went down to almost 29,000 leaves. So just a huge decrease for 1 year. And then the year after that, it had fallen even further to 26,500 leaves. The difference between 2023 and 2021, that's 24% loss in leaves just over the time that we were assessing the individuals at the site. The figure on the right, each of those dots is an individual across the 3 years. A significant difference between the first 2 years and then a decrease, but not a significant decrease between the second and third year. So the bigger difference was in the first 2 years. But if you look at the top of that plot, the individuals with lots of live leaves, you can see them dropping off. So these big individuals were just losing leaves slowly, and presumably also the smaller individuals. This other, sorry, that didn't really produce very well. But the next result I'm going to show you is the probability of individuals being infected across the 3 years based on how big they are. So that light grey colour at the top left, they're individuals that were never infected across the 3 years. Now, if you look at the bottom, you can see that they were always small. So it was only the small individuals that escaped infection entirely. And by the time you had a basal diameter, so if you measure the diameter of the plant base, as long as once you were greater than about a centimetre, so it's still pretty small, but once you're greater than a centimetre, you're almost guaranteed to have some level of infection. And then as the individual Lophomyrtus bullata individuals got larger, they're more likely to be infected across all of the 3 years, which is that darker grey colour. And the rest of the individuals who were infected either once or twice out of those 3 weren't strongly influenced by size. And we think this is because larger individuals have more leaves, more biomass, making them more likely to intercept spores and become infected. So this is a result from a Bayesian hierarchical model looking at the influence of forest edge on infection. So our main sort of question. The main response is the proportion of immature leaves in each of the 3 years of our study. I'm just showing you the first year at the moment, which is basically, the x-axis is showing the distance to edge, but it's been converted to a standardized effect size. So basically measure how influential the distance to edge is in the model, with positive numbers meaning distance to edge increases infection, and negative numbers, not that that's what we're seeing here in terms of our results, would mean distance to edge decreases infection. So I've added a red arrow to the plot and at the maps to help interpret this. So in the case of 2021, as you're heading into the forest, myrtle rust infections actually increases. The lines around the point show the critical intervals that Christy was sort of talking about before, sort of a measure of error. in the model, because with Bayesian models, you don't tend to talk about things being significant or non-significant, but you can kind of interpret those lines crossing zero as sort of a measure of significance. So in this case, we have a pretty clear effect in 2021 that disease increases as you head into the forest. So as I was sort of saying before, we were a bit perplexed by this at first. We expected there to be more rust at the edge of the forest, where the forest tends to be a bit more disturbed, less intact, maybe more entry points for spores. But sort of upon looking at the climate data, so this is relative humidity. This is the same kind of plot before. Positive numbers mean a strong influence of distance to edge on humidity in this case, and the different symbols of the different years. So 2021 at the top, 22, and then 2023. And this shows that relative humidity tends to increase as you get further from the edge. And this was consistent across all 3 years. Variable, but a consistent sort of positive, consistent positive trend there. And sort of suddenly it all sort of starts making sense. So if you look at this plot in the right corner there, that supports Rob Beresford's myrtle rust process model. It clearly shows, and Rob's been talking about this for years, that myrtle rust needs high humidity. You've sort of got this key humidity and temperature zone where you have higher infection risk of myrtle rust. So the next question, why was there increased humidity as you head further from the edge? We're not completely sure, but probably a range of reasons. It could be less wind, so more sort of stable conditions. But what we also found was this leaf area index, which I sort of mentioned before, was a sort of a measure of canopy cover. So as you get further in from the edge, you had increased canopy cover, so more closure of the canopy above you, which sort of makes sense when you think about it. It's getting darker, it's getting damper, more intact forests as you get further from the edge, which probably increased humidity and also myrtle rust infection. The influence of temperature was less clear. We found that temperature decreased actually as you headed in further from the forest edge, but we do know that the temperature was suitable across the whole time that we were there. So it was lower as you went in, but most likely still sort of suitable for the disease. Now, I sort of do need to say that with these plots, we're looking at how humidity changes across the edge, we're looking at how cover changes across the edge, and we're also looking at how disease changes across the edge. So all kind of correlative, not looking at the direct influence of those things on disease. When we look at the direct effects of humidity on disease and also on cover on disease, the results were less strong. The trends still came through. And there's a figure that I haven't got a figure here, but there's a figure in the paper that sort of shows that. So, and basically, I think that means that alongside humidity, there's a lot of other factors that change in this sort of complicated system. And we have limited kind of power with just our 3 trend sets, but I think that gives us a bit of a clue of what was sort of going on. So back to our annual assessments in 2022, our results still showed that infections increased as you got further from the edge. But you'll notice that the influence at edge this time is a little bit weaker. The point's a bit closer to 0. So we think we're starting to see this sort of result mediated over time. And then in 2023, the final year of monitoring the site a couple of years ago, the effective edge is almost gone entirely. So after 3 years of monitoring, and at least sort of 4 or 5 years of the disease being at the site, we believe that the disease has spread across the transects, including to the places that were initially less susceptible to infection, but still susceptible. So you're seeing the disease spread across the site. I'll finish just on a few slides of some other work that we've been doing at these transects. Some results that hopefully come out shortly. One is looking at these sort of inexpensive homemade spore traps, a cheaper version of what we heard about yesterday. We sort of got these fencing stakes and put these Perspex microscope slide holders, a little rain cover on top of them out in the field. And we, for a couple of days, I think we left these gel-covered slides out on them. And so these were the center of every one of those subplots that I was talking about before, to see how the level of inoculum changes across those forest edges using quantitative sort of PCR DNA quantitative technique. So those are some results that we'll be looking at, will be coming out shortly. And then the other, there's a microbiome study, of course. So the hypothesis being that myrtle rust infection will lead to decreased diversity of microbes, both at the site of the infection, so on the plant tissues, and also in the soil from surrounding plants. So from our 145 plants, there are about 90 that we decided could afford to donate a few leaves without dying to this project. And from each of those, we took a leaf wash to look at microbes on the surface of the leaves, and then we also looked at leaf tissues, so microbes from within. This came to a total of 720 samples, and we looked at 2 gene regions for that, and then in the soil from surrounding the plants, those same 90 plants, we looked at 5 gene regions. That is it for me. I'd like to thank Naruahine and Taranaki Iwi, who were both supportive of this work and came out with us a couple of times and helped. Landowners and DOC for site access, which was great. It was funded by Beyond Myrtle Rust, and there were, in addition to the authors on the paper, there were so many people that helped us through those years with sampling the site, through COVID restrictions and all sorts. So thank you very much.